Folks, we are bringing on the lovely Helen. So, Helen, welcome back, madame.
Thank you for having me. I'm honored, delighted.
Thank you for returning. We were so excited to have you on last time. I tell you, people were messaging me left, right, and center. When is she going to come back? When can we have her on? Can she be a regular? For those who are new, the uninitiated, who don't know the lore of Helen, would you mind a quick intro just to let folks know who you are, what you do?
Yeah, absolutely. Lore feels a bit extreme. I'm not as cool as Emily, who was just on here, but my name's Helen. I use she, her pronouns, and I work at Fred Hutch as their newest scientific content liaison. So essentially what that means is that I work in scientific education, outreach, and communication. And then before that, I worked in biotech research, studying cardiomyocytes or heart muscle cells and fibroblasts or muscle cells. And I'm also trained as a genetic counselor, which is a form of healthcare provider who helps folks understand their own DNA and use that information to empower them to make their own healthcare decisions. So that's me in a nutshell. Outside of science, I love soup and books and hanging out with my friends. So that's all I got.
What is the approach that we're taking today? Because I know we're doing something a little bit different from last time. What is the game that we're playing today?
Yeah. So what we're going to do this little game today, it's going to require participation from the beautiful and wonderful science community, as well as yourself. But essentially, I have put together a couple little medical mysteries, fascinating biological phenomena, chemical conundrums, RNA riddlers, helical whodunits. And what we're going to do is we're going to solve them together. We're going to learn a little bit about science along the way, and we're going to learn a little bit about some of the ongoing research at Fred Hutch.
All right, Daddy Uncle is our first case.
Our Fred Hutch patient, Vincent, was diagnosed with leukemia or blood cancer in his 50s. Which of the following do you think might be a helpful treatment for leukemia?
I mean, I'm feeling like it should be a combo, no?
Ooh. I mean, realistically, a lot of times, treatments for cancers are a combination, but for the sake of today, the one that we're definitely going with is a bone marrow transplant. And we call bone marrow transplants hematopoietic stem cell transplants because, we go back, we're learning this as a group. We have our person who's sick, they have bone cancer, or blood cancer rather. This means that these cancerous cells, these cells that grow too quickly, that outcompete your healthy cells, that go where they're not supposed to go in the body, that don't do their actual job of carrying blood and protecting our body as part of our immune system, these cancerous cells we want to get rid of. And so we're going to use chemo, we're going to use conditioning, we're going to do a variety of different things to prepare the body for a bone marrow transplant. And the reason this bone marrow transplant is curative for leukemia and so helpful in treating blood cancers is that what we do is we essentially wipe out a person's bone marrow ahead of time. So we get rid of their diseased, sick bone marrow, the bone marrow being that spongy tissue inside your long bones, your big bones, that makes all of the blood cell types you see on the screen there. So white blood cells, red blood cells, and platelets. We wipe out that sick bone marrow, and then we replace it with healthy bone marrow from a donor. And the reason it's also called hematopoietic stem cell transplant is because one of the really important cell types in that bone marrow is called a stem cell. And stem cells are just like little baby cells. They're capable of becoming any of those other cell types you see on the screen. So once you have a little baby stem cell, it can become a white blood cell or a red blood cell or a platelet. It has so much potential. We love it. And so because we love those and they're so important, we kind of have named these bone marrow transplants after them. And hematopoietic just means referring to your heme or your blood system.
And so there's this isn't like the pluripotent stem cell that can become a neuron, right? This is just like a earlier or later stage, but anything involved in your blood.
Exactly. And that's also why they're called hemopoietic stem cell transplants, because they're stem cells for the heme system, for blood system. But exactly like you said, they're not like totipotent or pluripotent. They can't become anything. They can become anything bloody.
Nice. Very cool. So that makes sense. So we're gonna put in this new replacement.
But if we go to the next slide, we'll get to our next question, which is where are we going to get that bone marrow from? It doesn't grow on trees. So in order to get a bone marrow transplant, Vincent's team at Fred Hutch first has to find a donor, someone to give them some bone marrow. So we've got a couple healthy volunteers. These folks raised their hand or paw and said, I will give my marrow. We've got his full brother, his cousin, his best friend, and his Labrador retriever. I'm curious who we think would be the best donor and why. When we think about bone marrow transplants, we want to match our donor to our patient. And the reason we want to do that match is because it's very important that the donor, the patient patient's body is able to accept the donor's bone marrow. So essentially, we want to trick our body, the patient's body, into thinking that the donor's bone marrow is their bone marrow. And that's because a huge part of the immune system is this ability to distinguish between self and not self.
I guess to pull back a smidge, so self tissue would be tissue that's inside of your body that belongs to you. Correct. And not self would be something that's at all foreign. So even if I get a splinter in, that's like non-self, something that the body could recognize as foreign and mount an immune response against it.
Right. And like when we think about what our immune system does, we often think of like our immune system being involved in fighting sicknesses. So your immune system is like, all right, this is Helen's skin cell, I'm not going to attack it. This is Helen's blood cell, I'm not going to attack it. But your immune system is trained to detect when something's a bacteria that we don't want in there, because we all know that we want some bacteria. When it's a virus, we don't want in there. When it's a fungus, we don't want in there. Or our immune system is even smart enough to detect when your cells are becoming cancerous, and it can kill precancerous and cancerous cells. What makes a cancer the kind of cancer you have to treat is when it's evolved and mutated in such a way where it can escape your immune system. Because exactly like you said, your immune system is trained to leave your healthy tissues alone and attack anything that isn't you or anything that isn't healthy. So what we do is we do our best to look at HLA markers as well as other, biological indicators of compatibility between your donor and your patient. We try to find a donor whose, blood and whose, organ looks the most like your own genetically and biologically. But that being said, we're also going to suppress your immune system because inevitably it's still a different person. And so we want to do our best to make it a hospitable, warm environment for that transplant.
And actually a cool question too from Hogan is an identical twin then the best match in terms of the genetics? Like if you had an identical twin, is that the best flavor of spare parts?
I do think that an identical genetic twin would be the best match genetically because they're going to have 100% of the same germline DNA, germline referring to DNA that you're born with. But again, you're still not going to be a perfect match because you might have a different microbiome, you might have different other biologic elements. But in terms of your actual DNA, that will be identical and identical.
Well, yeah, thinking of the microbiome as well, like that's a really complicating factor where if you're together, the two siblings, and they're living like apart from like the two different sides of the country, your DNA might match, but the gut microbiome, which would also affect things, is not going to match and is not going to have any chance to match. So HLA. Marker on the protein, kind of a signal that says, hey, I belong here or I don't belong here, right?
Exactly. If you go to the next slide, exactly what you just said. They serve as name tags for the immune system. So these HLA markers, they're like little flags sticking on the outside of almost all of the cells in your body. And what they do is they serve as name tags for the immune system. So your immune system, your immune cells float on through your body and they will notice these flags, these little name tags, and they'll read them out. And they'll say, that collection of HLA markers looks like something I'm comfortable with.
So my blood cells should not be in our patient or should not be in your bloodstream for the purpose of this, right? They're like, hey, this isn't no Helen right here. Get out of here.
Exactly. In this case, in this scenario, Helen and SciAnts are not compatible bone marrow transplants. And so if your blood cells got up in my body would be like, absolutely not. I don't want this bone marrow at all. This is terrible. I'm going to react as if it's a virus or a pathogen or something bad for me.
And that's even with the immune suppressants, right? There is just so much recognition of non-self that you're still going to have like a negative response in a way. Yeah.
Yep. Again, not a doctor, but the reason that we look for these matches and immunosuppression is that immunosuppression alone isn't enough to overcome an inherent mismatch. Mm-hmm. We can't just assume siblings are going to be a good match. And that's because there are many different types and subtypes of HLA markers, which are also different than blood types. So blood types, we think of O, A, B, AB. HLA is different. It's another confounding factor that's involved in these matches. But we know that there are a lot of different kinds of HLA, but that there are 5 that are most important for bone marrow transplants. And then a 6th one that they also think is pretty important.
Oh no, I was just curious, like these 6 different things that you have to genetically pre-screen the donor to make sure they're at least a match on 5 and preferably the 6th one as well.
Exactly. Yeah.
And so, and what is this, by the way, Helen? This image that we're looking at for the party people,
Yeah, this is a beautiful little chromosome. So, chromosome refers to a special little structure that lives inside your cells and holds your DNA. So, your DNA, which is just the genetic instruction set for your bodies, your DNA is long. There's so much of it. You are a complex, beautiful being, and there is a lot of instructions that your body needs to make you you. And so, in order to keep all that organized, we package that DNA into these structures called chromosomes. And this is just a little picture of where those genes live on one of those chromosomes.
So this is like a zoom in right here. So it's actually, they all live on this really, really tiny plot of land.
Yeah.
But we've just kind of blown it up. So these are the locations that we're looking for to see if we're going to get a match for the bone marrow in particular.
Correct. Yeah.
Okay.
So when we're looking for a bone marrow transplant, we're going to check these 5 genes and we're going to make sure they're a match. But not just everyone's a match. And that's because there are many different types and HLA genes are inherited. So if we go to the next slide, we remember that on the previous slide, we saw that zoom in.
Yep.
And there are these 5 different genes, these 6 different genes that matter. The cool thing is that they get inherited together, passed down from mom to baby and dad to baby, in chunks that we call haplotypes. Haplotype just referring to a chunk of genes that get inherited all together.
So we don't have to worry about those 5 being split and having some wild combination where you're trying to look for the clusters. So presumably if you test the patient, they're all 5 are going to be clustered together if they got it from mom or if they got their clusters from dad. Is that a right way of thinking of it?
Absolutely. And that like the clusters are consistent, but instead of like from mom or dad, folks get 1 from their mom or the egg and 1 from the sperm or their dad. So they have 2 total. They have, 2 chunks total, 1 from mom, 1 from dad. Sometimes you can see differences if during the formation of eggs and sperm, things got mixed up a little bit in a process we call meiotic recombination. Or sometimes we know that just changes can happen in genes. That's why not everyone's identical. So there can be instances where there are differences between parents and their child. But in general, exactly what you said. We inherit a chunk from our mom, we inherit a chunk from our dad. Those are our 2 chunks. We know that HLA is inherited. So a lot of times relatives are a good place to start when you're looking for a match. But there's only if you look, you see that for every set of siblings that share both parents, both biologic parents, there's a 25% chance there'll be a match. And that's because there are 4 different pairings. When you get 1 from mom, 1 from dad, you end up with 4 different possible pairings. But those are independent. So each child, it's a random roll of the dice, which of those 4 they're going to end up with. So each of your siblings, there's only a 25% chance you're going to be a match.
So the full brother may or may not be a match to you.
Exactly. There's a 25% chance the full brother's a match. That means there's a 75% chance that he's not a match. And we actually know that about 7 out of 10 people who need a bone marrow transplant can't find a match within their immediate family and rely on donors.
Okay, so our full brother answer may not be accurate. The numbers would actually go against it.
Yeah. We talked about how when you're matching HLA, there are a lot of different HLA genes, but there are like 5 that are really important. And you have 2 copies of each, 1 from mom, 1 from dad. And so you end up with a perfect match being called a 10 out of 10 match, meaning that each of those 5 HLA genes is a perfect match for both of your copies. And then if someone is even more well paired, like they have also like other HLA genes, including that 6th one I talked about, then they're called an above 10-10 match or beyond a 10-10.
And the higher the number, the more the preference is for that donor, right?
Exactly. I think, and again, not a doctor, not a bone marrow transplant specialist, but I think these days they really try and look for a 10-10 or above, because they also want to look for other factors like, is the donor relatively young? Ideally they want the donor to be relatively young, because we found that younger donors, young being under the age of 40, are more likely to have a successful transplant to a recipient, regardless of the age of the recipient.
Terminator Pig asked, so the odds that if you were to test your family or 100 random people, it would be more likely that it's the 100 random people that you'll get that higher tier match?
I think just because you're going with the numbers game, but in terms of like, if you have a random group of people and one of them is your full sibling or your parent or your child, there's going to be a greater chance you're going to be a match with a relative than a stranger. But if I had to pick between like testing just my sister or testing like 200 strangers, that might be a little bit, again, because there's only a 25% chance, 1 in 4 that my sister's a match. 75% chance, 3 in 4 she's not.
Mm-hmm.
But 200 people, maybe there's someone in there. We also know that it depends on the kind of people we're looking at, right? So We know that HLA, because it's inherited and genetic, often folks with similar ancestries are more likely to be matches to one another. So there's something that I'll get to later called the NMDP, or the National Marrow Donor Program, which actually started at Fred Hutch. But that's a registry that you can be part of where I did this recently. So what you do is they send you a little kit. You do a little cheek swab kit. You send back your little swabs with your cheek cells, and you send that back to the lab. And then they will look at your HLA and like your general match. They'll put that in a big old registry. And then when someone needs a bone marrow transplant and they can't find a match, they check that list and they go, is someone on here a good match? Jackie from Fred Hutch, she's also on it. I'm on it. And like, you could save someone's life. Like if you're a match for someone, you can give them your stem cells. You can give them your bone marrow. You can be their donor, which is just like so cool. Vincent's brother, Anton, was able to make a successful donation, and Vincent is now in remission, which we love to see. But for the holidays this year, Anton thought it would be fun for them to all do one of those like direct to consumer ancestry kits, like 23andMe or Ancestry. And so he ordered kits for Vincent, himself, Vincent's daughter, his own son. And they all sent in their cheek swab samples, and they just got back the results. So if we go to the next slide and we think about everything that we've talked about so far, what surprising thing do you think those test results said about their familial relationships? It came back saying that they're identical twins, which is shocking. Do we know why? Like why the test would say that? So this is like a legit thing. And this is actually based on like a real life situation. So what happened was the test, first of all, says that Anton and Vincent are identical twins. And that's because we know that Vincent now has Anton's bone marrow. And what they did was they did a cheek swab sample. So if we think about what kind of cells are included in a cheek swab, that's going to be white blood cells, and that's going to be epithelial cells. So epithelial meaning those skin cells inside of your mouth. And it's the white blood cells that really mess up with that sample, because the white blood cells come from the bone marrow. And so those kind of mess with the sample purity. And by looking at the DNA in those white blood cells, they came back saying that, this is actually Anton's DNA. Vincent and Anton must be identical twins. And therefore, they also showed that Anton's son was Vincent's son, and that Vincent's daughter was like Vincent's niece, because his DNA is not the same as Anton's. And this catchphrase, when in doubt, punch it out, means if we don't know if this person has a blood cancer or some sort of hematologic or blood-based disorder, if you want to make sure you're getting the DNA that person was born with and not the DNA from the cancer, because cancer DNA is always messed up, because we know that cancer, as we talked about last time, is always genetic, but not always hereditary. We know that if we get a skin punch biopsy, that's going to be the DNA that person was born with, because very little, aside from like actually replacing a person's skin with someone else's skin, is going to change the DNA in your skin and muscle. So when in doubt, punch it out, meaning when in doubt, get a skin punch biopsy instead of taking a blood sample or a cheek swab.
And that should catch the cell populations that are not affected by the bone marrow transplant.
Exactly. Yeah. We were talking about whether or not the people here might be interested in the microbiome.
So y'all, I may have suggested that y'all might have been interested in the gut microbiome, because we've had a lot of questions either on these kinds of collabs or just on Science News Fridays, or just like a lot of different instances where it's like, all right, what is the connection to the gut microbiome and this phenotype, right? Look, there we go. Look at that. Hell, did I not say the comments of I love microbiome? Where else are you going to find that on Twitch?
No, you guys, the microbiome is the coolest, and it's crazy because like Jackie and I were talking about this the other day, because we attended a talk with, not attended, Jackie, being the brilliant, beautiful person she is, allowed me to help her with an event that she was putting on, featuring some nutritionists talking about sort of diet and like the ways that we can help reduce our risk for cancer by altering sort of certain modifiable lifestyle factors, which again, I always include with an asterisk, because like just because something is modifiable doesn't always mean it's actually within your power at that moment to change. But we were talking about how diet can influence things. And Jack and I were both freaking out about this thing that one of the nutritionists said, where she was like, you need to not just feed yourself, you need to feed your microbiome. Because like, and it made me think like, yeah, like I need to feed my microbiome. Like I need to make sure that like she's getting what she needs. There I go, gendering it for no reason. But like, like you need to have macros, because like the bacteria need protein, but the bacteria need roughage, and the bacteria need prebiotics and probiotics. And you need to have balance and diversity, because different bacteria want different things. And if you are too restrictive with your diet, then certain bacterial species are going to overgrow and others are going to die out. And like, that's just crazy. All that being said, since some of you were into the microbiome, and I guess I can clearly tolerate talking about it. If we go to the next slide, have a connection. Okay, so what they're finding is that the microbial genes, the genetics of your microbiome, are non-trivial when it comes to bone marrow transplant. And that's because bone marrow transplants don't just introduce new cells, they also bring in new immune cells. So when I donate that bone marrow, you're donating those hematopoietic stem cells, which can become the red blood cells, the white blood cells, and the platelets, but you're also donating existing white blood cells, cells that ideally can kill the cancer cells that are still in the patient. Because remember, that's what our T cells are trained to do. They're trained to kill invaders and also cancer cells. So you see in green, I've got this GVL that's called graft versus leukemia. And that's when the donor's immune system kills cancer cells in the patient, which is super cool. And then you also have GVHD on the other side, which is bad. And that's when it's graft versus host disease. And that's when the donated immune cells start to kill healthy cells in the patient. And we don't want that. We do want them to kill the cancer cells, GVL. We don't want them to kill healthy cells, GVHD. And we also don't want rejection, which is when the host's immune system attacks the donation. And that's why there's that line of like, we want the body to think that it's their bone marrow and their immune cells, so you don't get GVHD or rejection. But you also do want those immune cells to still work. So you don't want the immune cells to be so deadened or absent that they don't still attack cancer cells, because you still want them to attack cancer cells. And then after this person's healed, you still want them to have some level of immune system, right? Like you still want them to be able to fight the common cold.
So you're like your everyday infections, you still want those to be functional.
Exactly. Like if I cut myself, I don't want to need to get a tetanus shot because I'm, peeling a sweet potato aggressively. But we've got some amazing scientists who are currently studying this specific problem, figuring out that our microbiome is involved in bone marrow transplant. And we've known that for a while, but we're trying to figure out how. Like they knew the microbiome was involved in bone marrow transplant success and graft versus host disease, but they didn't know exactly why. They just knew that it made a difference. And so they were doing this study in mice, and what they found is that the donor's immune cells, specifically T cells, which are kind of white blood cell, interact with the recipient's microbiome, and that they can exacerbate GVHD. And if we can learn how to identify which of those T cells, because we know we have a lot of different kinds of T cells, different populations. It's not like you have one kind of T cell. We all have many different kinds, different T cells for fighting different things. We know that if we can find a way to identify which T cells are responding to the bacteria and making GVHD worse, we can remove those and leave behind the T cells that are fighting the cancer cells and not impacting your healthy tissues.
So selectively getting rid of the bad cells that you don't want and leaving the good ones.
Yeah.
With the gut microbiome like at play, like that's crazy.
Yeah. This is just kind of going through like the way they figured this out. So essentially what they did was this white mouse that I have here on the far left. That's our bone marrow donor. And they took bone marrow from this mouse and they put it into these two genetically identical mice. And they were like, all right, if only the genetics of the mouse matter, if only the innate germline genetics the person is born with matter, then they should respond exactly the same to this bone marrow transplant. And they should grow up the exact same populations of T cells. Because remember, T cells, like your microbiome, are unique and diverse and beautiful, and there are so many of them. And they vary from person to person depending on a variety of factors. And we think some of those factors are your microbiome. Because they found that these genetically identical mice that had distinct and unique microbiomes also had distinct and unique populations of T cells. So they might have some overlapping T cells that are the same, but the ratios were different. Does that make sense? Mm-hmm. And there were some unique ones.
So if it was just the genetics, so the two gray mice, y'all, are genetically identical. Yeah. And if they're getting this transplant, if it's just genetics that matter, all the outcomes should be the same. And that's not what they find.
No, that's not what they find. So this is another thing they did. So they were like, okay, so we know that the microbiome is affecting things. We know that it's involved. So like, how do your T cells, which are an immune cell, even know like that the microbiome is there? Like how are they even interacting with them? And they think it's through this something called the TCR, or the T cell receptor. So our T cells have these little receptors all over them. And these receptors are essentially what allow them to interact with other cells. HLA is actually involved in that TCR. This is an antigen presenting cell, also called an APC. So this is a cell with some kind of HLA marker on it. So I'm a cell and I've got an HLA marker. And the HLA marker is a protein that conveys information about what I'm like. And specifically right now, my HLA is also taking a little piece of protein from inside the cell and holding it very delicately between its tiny little HLA hands. And this is a way for my immune system to learn about not just what the cell is based on the name tag, but also what's going on inside. Because now my name tag has like affixed a little piece of what's inside me on the outside of me. Because the analogy is breaking down. And then my T cell has something called a TCR, or T cell receptor, which goes in and reads that HLA molecule. And in that way, they're able to interact. Did I represent that in a way that made any sense at all?
It's like the interaction is based on what the markers are, if they can interact or not.
Yes. And it's also based on what's inside the cell. So TCRs are a way that we can use TCRs or T cell receptors to interact with the inside of a cell and not just stuff that's already expressed on the outside. And we're actually using TCRs as a form of chemotherapy now. Not chemotherapy. And so what they did was they engineered these TCRs, put them on the T cells, put that in the bone marrow. And then they put that in two different genetically identical mice. And one of those mice, the top mouse, had way more bacteria than the bottom mouse, which had very little bacteria. And so then they observed what kind of T cells grew up in those two different mice. And they found that the mouse with more bacteria had T cells with more TCRs specific to the bacteria that was engineered, and had worse graft versus host disease, meaning that the bacteria are interacting with T cells via the TCRs to worsen versus host disease, which is crazy.
That is really cool. Wow. Yeah.
So they're finding that the microbial genes are involved in mice. But like, does this hold true in people? We're pretty sure, but we need to do some clinical trials to figure that out. Which is another thing that's cool about Fred Hutch, is that we have so many clinical trials, which are a way to increase access to leading edge treatments for folks who might not otherwise be able to access them, and to make sure that our treatments are as safe as possible, and that people are always getting the best care and not just the standard of care.
Mm-hmm.
And the other thing is that if we know T cells responding to some bacteria makes graft versus host disease worse, presumably they can respond to some bacteria in a way that could actually be protective and like make it less likely that graft versus host disease can develop. So right now they're trying to figure out like if some bacteria helps you.
So that's almost like the question earlier tonight with like there are viruses that could also be helping you, right? But here there's like actual example of the bacteria doing the bettering and actually helping.
Exactly. Yeah. And so then if we go to the very next slide, we have two clinical trials. And this is the last one for this case. And then I'll give you a sneak peek of case 002. But we have two clinical trials that are really exciting that involve basically doing a transplant of one person's microbiome into another person and then seeing if that affects graft versus host disease. One of the other things I like about Fred Hutch is that we have so many projects that are focused on learning more about the diversity of humanity, because no one person is alike, and we're all alarmingly alike. And you have to be able to hold both of those truths in your mind at the same time, and you have to respect them equally, because without fully believing in and comprehending both of those ideas, that we're all fundamentally strangers to one another and we're all fundamentally identical, without having both, you'll never be able to treat cancer effectively. And so I think it's crazy that I've found a workplace where like everyone believes those things, because like those are crazy things.
It is really, really cool. Yeah, 100%.
Yeah.
All right, so that wraps up this first case. We're going to let Helen go so she can have dinner.
Thank you for having me. Always a pleasure. Thank you all. Congratulations to everyone who won things. Thank you all for donating. Thank you all for listening, asking questions, participating. And of course, thank you to SciAnts for hosting us always so graciously.
Thank you for your time, your expertise, and your constant undying support from you, Jackie, and all of the Fred Hutch team. It is incredible. Thank you so very much for coming on with us tonight.
Thank you. Thank you all. Have a good night.
Bye, Helen.
Bye.